Good morning, students. In the today's lecture, we are going to discuss about the life cycle of the Cercospora. Okay, today's topic for the discussion is the life cycle of the Cercospora. First of all, the systematic position or the classification of the Cercospora. Here we are following the classification which was proposed by the Alexopoulos and the Mims in the 1979. 1979. Okay, as per the classification proposed by the Alexopoulos and the Mims in the 1979, all the fungi these are placed in one kingdom, and that is the kingdom Mycete. Here you can see the Cercospora. This fungi it is also placed in the kingdom Mycete. Okay, it is placed in the kingdom Mycete. Then division Amastigomycota. Amastigomycota, that is the division in which there is the fungi which are not producing the any motile cells during their life cycle, or at any stage of their life cycle. All these fungi are placed. That means the Cercospora it is not producing the any type of the motile cell at any stage of the life cycle. Okay, kingdom Mycete, division Amastigomycota, then subdivision Deuteromycotina, form class Hypomycetes, form order Moniliales, then form family Dimaceae, and the genus is the Cercospora. Okay, here the classification of the Cercospora: kingdom Mycete, division Amastigomycota, subdivision Deuteromycotina, form class Hypomycetes, form order Moniliales, form order Dimaceae, and the genus is the Cercospora. Okay, in this classification here you can see the terms like the form class, then the form order, and the form family. Okay, the question may be arised, why the this classification it is using the terms like the form class, form order, and the form family. Okay, because the classification of the Deuteromycotina it is artificial, hence the various taxa are known as the form order, form family, and the form genus, etc. Okay, the classification of the Deuteromycotina it is artificial, and hence the various taxa these are known as the form order, form family, and the form genus. Okay, why we are saying that is the classification of the Deuteromycotina it is artificial? Because the subdivision Deuteromycotina includes all those fungi in which only the asexual or the imperfect stages are known, and the sexual or the perfect stages these are not yet recorded, these are not yet reported. Okay, the asexual reproduction it is said to be the imperfect stage, and the sexual reproduction in the fungi it is called as the perfect stage. And the subdivision Deuteromycotina it is said to be the artificial group, because it includes those fungi in which only the asexual or the imperfect stages are known, and the sexual or the perfect stages are not yet recorded. And therefore the members of the Deuteromycotina these are popularly called as the fungi imperfecti. Okay, the members of the Deuteromycotina these are popularly called as the fungi imperfecti due to the absence of the perfect stage, that is the sexual reproduction. Okay, these fungi as such this cannot be placed in the well characterized groups, that is the well characterized subdivisions of the Mastigomycotina, like the Zygomycotina, then Ascomycotina, and Basidiomycotina. And also these cannot be placed in the division Mastigomycota. Okay, remember that these fungi as such cannot be placed in the well characterized groups like the Mastigomycotina, Zygomycotina, Ascomycotina, and the Basidiomycotina. Okay, this subdivision Deuteromycotina it includes about 600 genera and over 20,000 species. Okay, the subdivision Deuteromycotina it includes about 600 genera and over 20,000 species. They show the resemblances with the members of the Ascomycotina and the Basidiomycotina. Okay, these are showing the resemblance with the members of the Ascomycotina. Okay, we have discussed the member of Ascomycotina, that is the Aspergillus, and also we have discussed the member of the Basidiomycotina, that is the Agaricus. Okay, and the fungi which are included in the Deuteromycotina, these are showing the resemblance with the members of the Ascomycotina and the Basidiomycotina in some features. Okay, the several members which were previously included in the Deuteromycotina, due to the absence of the perfect stage, that is the sexual reproduction, but later on these were transferred to an appropriate group when their perfect stages were recorded in nature or in artificial culture media. Okay, remember that the several members which were previously included in the Deuteromycotina, due to the absence of the sexual or the perfect stages, but later on these fungi were transferred to the appropriate group when their perfect stages were recorded in nature or in the artificial culture media. Okay, and thus we can say that the subdivision Deuteromycotina, it is the purely artificial and the temporary assemblage of the fungal species waiting to be included in an appropriate group after the discovery of their perfect stages. Okay, thus the subdivision Deuteromycotina, it is purely the artificial and temporary assemblage of the fungal species waiting to be included in an appropriate group after the discovery of their perfect stages. They are defined as the conidial stages of Ascomycotina, or more rarely of the Basidiomycotina, whose sexual stages have either not been discovered or do not exist at all. Okay, these fungi, these can be defined as the conidial stages of the Ascomycotina, or more rarely of the Basidiomycotina, whose sexual stages have either not been discovered or do not exist at all. Okay, that is regarding the subdivision Deuteromycotina, which is popularly called as the fungi imperfecta, and it is the artificial group. Okay, and that is all about the classification of the Cercospora. Okay, here in this lecture we are going to discuss the member of the Deuteromycotina, and that is the form genus Cercospora. Okay, here the first of all habitat of the Cercospora. Okay, Cercospora, it is a very large genus of the family Dimaceaceae, and it includes about more than 3,800 form species. Okay, Cercospora, it is a very large genus of the family Dimaceaceae, and it includes about 3,800 form species. Majority of the species, these are parasitic, and these are responsible for the leaf spot or the ticca disease of economically important plants. Okay, majority species of the Cercospora, that is, these are parasitic, and these are infecting the plants, and these are responsible for the disease which is commonly called as the leaf spot disease or the ticca disease of the economically important plants. Okay, one of the species of the Cercospora, that is the Cercospora epi, it is the human pathogen. That means it is infecting the human beings, and it may cause the several lesions on the face. Okay, thus remember here the Cercospora, it is the parasitic fungi. It is the large genus of the family Dimaceaceae, and of which that is about 3,800 species. These are reported on all over the world. That is about the habitat. And now here you can see the symptoms when the Cercospora, it is infecting the certain plants. The fungus, it mainly infects the host leaves. Okay, the Cercospora, it infects usually the host leaves, but when the infection is severe, the stem is also get infected. The infection, it begins as the pale green spots on the upper surface of the leaf. Okay, the infection of the this Cercospora fungi in the plant, it can be characterized by the presence of the pale green spots on the upper surface of the leaf. Okay, but these spots gradually get enlarged in the size, and they will turn brown in color, and ultimately the entire leaf dries and crumble down. Okay, here in this photograph you can see that is there is a formation of the brown colored spots on the upper surface of the leaf, and this is the ticca disease of the groundnut. Okay, the leaves or the plant which is shown in the photograph, it is of the groundnut, and the disease, it is called as the ticca disease of the groundnut. Okay, ticca disease of the groundnut. Okay, here these spots gradually enlarges in the size, then these spots turn brown in color, and ultimately the entire leaf dries and crumbles down. Okay, here you can see the some of the important diseases which are caused by the different species of the Cercospora. Okay, it is the summary of the diseases which are caused by the Cercospora to the some of the economically important plant. Here you can see the leaf spot or the ticca disease of the groundnut, which is caused by the species Cercospora arachidicola and Cercospora personata. Okay, then Sigatoka leaf spot disease of banana, which is caused by the Cercospora muci. Then frog eye leaf spot of tobacco, that is the Cercospora nicotiani. Then leaf spot disease of the lady's finger, that is the Cercospora hibiscus. Then Cercospora indica, it is responsible for the leaf spot disease of the Cajanus cajan. Then Cercospora beticola, it is responsible for the leaf spot disease of the sugar beet. Then Cercospora oryzae, it is responsible for the leaf spot disease of the paddy crops, for example, rice. Okay, Cercospora oryzae, it is responsible for the leaf spot disease of the paddy crops. Then Cercospora gossipina, it is responsible for the leaf spot disease of the cotton. Here, these are the some of the examples of the diseases which are caused due to the different species of the Cercospora. Okay, from this it is clear that the Cercospora, it is mainly infecting the economically important plants, and it usually attacks the leaves of the economically important plants. Then here the next one, that is about the vegetative structure. About the vegetative structure, here you can see the mycelium, it is profusely branched. The mycelium, it is profusely branched, it is hyaline and septate. Okay, in this photograph you can see that is the mycelium, or it is the single hyphae of the Cercospora, and it is a septate hyphae. Septate means here the transverse septa or the partition walls are present. Okay, the mycelium, it is profusely branched, it is hyaline and septate. It is both the external and the internal. The hyphae grows in between the intercellular spaces of the host tissues and derive the food materials with the help of the branched hostoria. Okay, these hyphae, these are developing the hostoria to absorb the food material from the host tissue. And these myceliums, these are present in between the intercellular spaces of the host tissues. Okay, then the that is about the structure of the vegetative mycelium in the Cercospora. And here this is the photograph, that is which are showing the leaf spot diseases of the groundnut. Okay, this is the groundnut plant, and here the Cercospora, it is infecting the leaves of the groundnut plant. And here you can see the leaf spots which are developed on the upper surface of the leaves. Okay, that is all about the vegetative structure in the Cercospora. Then regarding the reproduction, just we have discussed that the Cercospora, it reproduces only by the asexual reproduction by the formation of the conidia. And the sexual reproduction, it is either absent or it is not reported yet. Okay, or there may be the complete absence of the sexual reproduction in the Cercospora, and that's why it is placed in the artificial subdivision, that is the Deuteromycotina. Okay, reproduction in the Cercospora, it reproduces only asexually by means of conidia, which are developed on the long slender conidiophores. Okay, here you can see these long slender structures which are called as the conidiophores, and on these conidiophore, at the tip region of the conidiophore, there is a presence of the conidia. Okay, at the time of the formation of the conidia, hyphae accumulates just below the epidermis of the host and form a compact brown or the black pseudoparenchymatous mass, which is known as the stroma. Okay, here you can see this is the epidermis of the host tissue, and just below the epidermis, the large amount of mycelium of the Cercospora, it get accumulates and it is forming the pseudoparenchymatous like structure. Okay, pseudoparenchymatous like structure, and that pseudoparenchymatic structure of the mycelium of the Cercospora, it is referred as the stroma. Okay, here easily you can see that is these mycelium of the Cercospora, that is these are growing in the intercellular spaces in between the host cells. Okay, and just below the epidermis, a tuft of short septate thin walled and unbranched conidiophores emerges out from the stroma. Okay, here you can see from the stroma, that is there from the stroma there arises the thin walled, then septate and unbranched conidiophores. Okay, from the stroma then arises the thin-walled unbranched conidiophores. Each conidiophore, it produces the conidium at its apex. Okay, here in this photograph you can see at the tip region of the conidiophore there is a presence of the conidium. Okay, the first former conidium is thus pushed to the one side, and another conidium it is formed at the apex side. Okay, then this the process it may repeat several times, and as such the several conidia these are formed on the conidiophore. The conidium here, it is the structure of the conidium. Okay, the conidium here you can see it is a straight or curved. Okay, the conidium which are developed in the Cercospora, these are straight or curved, these are hyaline, or these are pigmented. Then these conidia, these are slender, narrow, tapering and transversely septate. Okay, the conidium which are developed during the asexual reproduction in the Cercospora, these are straight or curved, these are hyaline or these are pigmented, these are slender, then conidia these are narrow, tapering and transversely septate. On detachment from the conidiophore, the conidium leaves a small scur at the place of its attachment. The conidia, these are dispersed by the wind or the rain splash. Okay, these conidia, these are get separated from the conidiophores, and then these conidia, these are dispersed by the wind or the rain splash. They germinate under the favorable conditions by giving rise to one or more germ tubes. Okay, whenever the conditions are favorable, then at that time these conidia they will germinate and they will form one or more germ tubes. Each of the germ tube it ultimately develops into the new mycelium. Okay, each of the germ tube ultimately develops into the new mycelium. The optimum temperature for the growth of the Cercospora, it is between the 24 to 28 degrees centigrade. Okay, optimum temperature for the growth of the Cercospora mycelium, it is about 24 to 28 degrees centigrade. Okay, the in the some of the form species of the Cercospora, the perfect or the sexual stages these are reported, and okay, but in most of the species, that is the sexual reproduction, it is not reported. Okay, that is all about the asexual reproduction in the Cercospora. Okay, and that is all about the life cycle of the Cercospora.